Okay, guys, let's start. Today we'll be doing an analysis for Term 2 STPM Biology. All right. So first up, let me share screen. As for the handout, I think I will pin the number. I'll put it somewhere so that if you want the analysis for the past 10 years, you can get it from my staff. Just send a WhatsApp and then they'll WhatsApp to you. Okay, so today's one is supposed to be a short session, but after I prep for this video recording, I feel it'll be quite a while as well lah. Actually, my target was 10 minutes, but I think looking at what we have here, maybe it'll be slightly more. Now, first up, it depends on where you're watching this because I'm recording it today and uploading it today as well. Guys, we are here. Today is the 6th of August. That's exactly another one more week before you fight in Sem 2. Okay, your next battle. Normally, Sem 1, I call it Battle 1. Sem 2, Battle 2. Then Sem 3 is the war. So how come both are, all the three are paper 1, paper 2, paper 3, they are kind of the same thing, but then you call it battle, and the last one war is because the last one you cannot retake. And you retake Sem 1 and Sem 2 around Sem 3. That's why I call it a war for the last one. Right. So 1 week before your STPM, guys, a few pointers for bio. Not any other subjects. Number one, I think that at this point in time, you have done most of what you should already plan to. Okay, by right, whatever should be done has been done at this point in time. Normally, the last week is to touch up, make sure things are all right before you go into the exam hall. But just to make sure things are done right, number one, you should have covered all the subtopics. Guys, you should have covered all the subtopics. Meaning, if you still haven't yet, you still have time. So with the remaining time, if you haven't read everything, don't go and try state papers or stuff like that. Guys, you need to always have a foundation before you try questions. Number one, you have covered all the subtopics. Number two, 2 days before your bio, or maybe a day before, you browse through all the questions that you did, all the questions attempted before. Okay, soalan yang Anda pernah buat, just go through the question and answer. Guys, again, I'm not asking you to try new questions. Number three, avoid new questions a day before exam. Because confidence matters. Guys, confidence matters. Meaning to say that one day, if you haven't been trying much questions and you suddenly try many questions a day before your bio, you are essentially killing your own confidence. Never ever do that. Yeah, so if I'm being very upfront, guys, you had 6 months to do what you needed to. Why are you doing it 1 day before? Okay. So I'll leave this to you. Okay, I'll leave this to you. Now let's look at the analysis, right? So as I mentioned just now, you may obtain this from my staff. You just have to WhatsApp the contact that I'm going to include. All right. So how we're going to run the session today is probably you need an empty A4 paper. And if you have this printed already, it's good to have highlighter. Okay, it's good to have highlighter. Now, talking about this semester's exam, Sem 2, particularly for now, meaning 2024, not the past 1-2 years, because whatever I said for last year is not applicable for this year. Apa yang telah keluar takkan 90% takkan keluar dalam SEM 2 peperiksaan SEM 2 ini. Yes? Okay. A few things to note. Number one, why this analysis is important is because 90% of whatever asked last semester will not come out. Okay, 90% of whatever asked in the last semester will not come out. I just show you a very brief example. Guys, I just show you a very brief example. Just browse through non-steroid hormone, blood glucose, control of breathing, stomata, okay, human heart, pathway of water, the dengue. Just check the year before. Cardiac cycle, nephron, body temperature, immunity response, gibberellin. Guys, if you check until the end, nearly nothing came out. Nearly nothing came out. Then if you take this year compared to the previous year, gut cell, sliding filament theory, okay, heart sound. So if you check year after year, likelihood of a repeated question is less than 10%. So has it happened before? Definitely, definitely. Okay, look, I just show you one. Where's the structure? Where's the structure? Guys, look here. Breeding cycle, 2014, 2015, when they started the new format back then. Okay, which comes to the next part. So, how do they ask the 10%? There are a few ways. The less than 10% chances of whatever came out last year is from structure, they switch to essay, or essay, they switch to structure. Number one. Number two, guys, they ask the same subtopic yang sama, but different, totally different questions. Totally different questions, okay? And with that said, guys, whatever came out last year should be kind of like the last thing for you to study. The last one on your list. I'm not saying don't study, yeah? Please don't get me wrong, because this is SEM 2. And what's the challenge with SEM 2? It's very much heavier than SEM 1 and SEM 3. Wow, sir, are you saying that SEM 1, 2, 3, the heaviest is really SEM 2? Yes. Yes. Okay. And the third thing, guys, topics that are more important this semester include 8, 9, 10. 8, 9, 10. Yeah, 8, 9, 10. Later you'll see. Reason is because SEM 2 is a very heavy semester for bio. I always tell students, SEM 1 is easy. Yeah, for bio, SEM 1 is easy. SEM 2 is long. SEM 3 is hard. SEM 3 is hard. But whenever I say SEM 2 is long, SEM 3 is hard, long and hard, then students, the boys will keep laughing. I don't know what nonsense they're thinking about. Okay, anyways, guys, so the topic to really take note for your SEM is chapter 8 to 10. Chapter 8 to 10. Okay. Oh, by the way, I have the habit of seeing it as 7 to 13. Because to me, it signifies progress. Kamu telah ambil peperiksaan SEM 1 di mana ia melitupi bab 1 sampai 6. So lepas tu next part is 7 to 13. Is to let you know that you have finished one semester, you are here, then you're halfway to the end. Yeah. So after this semester, it's good to have a breather, reflect, and then continue. Okay, guys, look. Let's look at the analysis straight. I'll be using, let's say, a colored pen. Let's say we go for this maroon-ish color. For chapter 7, what came out last year is control of breathing mechanism and also response of stomata. The previous year, they didn't ask anything. Of course, you can see gut cells here. Guys, you can see gut cells here. And then 2020 don't have oxygen dissociation curve. Guys, oxygen dissociation curve. Okay, you can continue to move backwards if you want. Now, this batch, okay, I just want to add one more sentence. This is for students who fail their bio lah. This is for students who fail their bio. Probably for students who fail their bio, okay, probably what you can do is you don't bother about what came out the last 2 years. Don't bother about what came out the last 2 years. Because if you fail your bio means you're obviously not there yet. Fail your percubaan. So this is to help you lighten your burden a little. Yeah. Okay, look. So in the context of chapter 7, the most important part to me is this. It's been a long time since they asked this part. Transport of CO2, meaning are you able to explain? Are you able to explain transport of carbon dioxide? Let's say if I were to go through a very quick one with you. Guys, normally in class, I'll use PowerPoint because it's more efficient. Instead of, whiteboard normally is to discuss analysis. Okay, so let's say we are looking at chapter 7 here. Let me include the audio. Okay, guys, look. Oh, normally I have to censor this. Yeah, I have to censor this. Okay. See, this one I'll censor. Hey guys, one last time, this one I'll always censor. Yeah. All right. So we are studying how CO2 move into the blood capillaries. If you're using visual like what I'm doing here, your revision should be very fast. Look, partial pressure of CO2 in body cells is high. CO2 will diffuse into the blood capillaries. Guys, as you already know, there are 3 forms of how CO2 is in the blood capillary. About 7% will move into the blood plasma as dissolved CO2 gas. About 23% enter red blood cell combined with hemoglobin. Revision, what is formed? The answer should be carbaminohemoglobin. Remaining 70% enter red blood cell combined with water to form carbonic acid. Guys, recall what's the enzyme? At this point in time, you must already know. Okay, this is carbonic anhydrase. Carbonic acid will split into hydrogen ion, bicarbonate ion. Guys, what happens to bicarbonate ion? Because you're doing revision, you have to be one step ahead. It will diffuse into blood plasma. And what's the next step? Chloride shift to maintain electrical neutrality. Finally, hydrogen ion combined with oxyhemoglobin to release the oxygen. Okay? Hemoglobinic acid is formed. Oxygen will be diffused into the body cells. Okay, guys, one more round. I just want to show you one thing. One more round. Partial pressure CO2 high, CO2 diffuse into blood capillaries. About 7% are dissolved CO2 gas. About 23% enter red blood cell, react with hemoglobin, form carbaminohemoglobin. 70% carbonic acid. Okay, carbonic anhydrase, this one dissociate. Bicarbonate ion diffuse out into blood plasma. Chloride shift, maintain electrical neutrality. Then hemoglobinic acid is formed. Guys, what I'm trying to tell you is, if your revision has been solid, you can revise the whole thing in 1 minute. If you are able to revise processes in 1 minute, you are top 1% in Malaysia. Definitely top 1% in Malaysia. Malaysia looks a bit funny today. Oh, sorry, top 1% for this process, not for the whole bio. Because at the end of the day, SEM 2 are full and packed of processes. Yeah. Okay, next. Let's move back to the chart. So guys, the first most important thing is that this part is more important, and I just showed you roughly how you should revise if got process. Okay, number one. Number two, if they want to ask you, so this I'll put it 2 stars. This is quite important. If they want to ask you trick question or surprise question, like surprise, you never expect this to come out, it would be characteristics. It would be hemoglobin, guys. This part. Structure and characteristic of hemoglobin. If not, structure and function of stomata. So for this topic, please cover these few things before you cover the rest. In fact, what I would recommend you to do is when you revise all this important part first, then we come to the rest later. Okay, of course, this one already came out last sem. Next, chapter 8. Okay, chapter 8, this is the mind map. This is the mind map. Let's check out what came out the past few years. Okay. I'll change to another color. Let's say blue color. Let's say blue color. Last year, you can see here that they asked about 3 pathways of water as well as how blood move in the correct direction. Okay, cardiac cycle the previous year, then heart sound, disease, arteriosclerosis, transpirational pool. Okay. So for your semester, guys, the more important part, because there are many things to study in Bug 8, 9, 10, like it or not, you have to put in more effort. The focus is still a bit more to blood circulatory system. Guys, the focus is still a bit more to blood circulatory system. Oh, let me add in the lymphatic system so that it's complete for you. One sec. Okay, done. I just added this. For lymphatic system, right? Lymphatic system, I mean, of course, you can take note of the calculation, but tissue fluid formation is very simple by now. I feel it shouldn't be a problem because since Form 5 since Form 4 you've been learning. Oh, blood entering arterial is under high pressure. Due to this high pressure, blood plasma is forced out to form the tissue fluid. Last time we also learned it as interstitial fluid. Okay. Then as for function, you can just study it in chapter 11, immunity. So to me, lymphatic system here, as long as you know the calculation, you know how to calculate filtration rate, absorption rate, then you are very safe. Then you are very safe. There are 3 methods, guys, to calculate. Sorry, there are three methods of, not 3 method of calculation. I don't want to confuse you. 3 methods of how questions are asked. Yeah, but I think your teacher would have covered that. That one is quite basic. Okay, so coming back to blood circulatory system, guys, coming back to blood circulatory system, definitely most of the things you have to really spend time to go through. So how I would position the whole thing is everything under blood circulatory system as well as translocation is your top priority. Guys, top priority. Okay, because if you look at it, like I said, because the content of the topics are very heavy, there are still a lot of things that they can ask. Is it cardiac cycle? Maybe cardiac cycle is of lesser importance. Maybe cardiac cycle is of lesser importance. Okay, so I just want you to, now the thing is this, you don't have to really spend a lot of time as well. Okay, this one is of lower importance. Heart structure, I think is fine. So maybe if you really want me to scope it to a few things, it would be 1, 2, 3, 4. Guys, initiation of heartbeat and control of heartbeat are two different processes because initiation is pumping of heart is myogenic in nature. It starts at the SA node. What SA node is generate electrical impulse. Electric will be sent to the walls of atria. Walls of atria, then after that atrial systole, then you continue a few points. The last point is always heart diastole. Guys, revision is actually less than 1 minute if you know how to go about it. Whereas this one here involve the nervous system. That's why the syllabus they put there nervous control of heartbeat. So this is for blood circulatory system. Guys, translocation, of course, number 1 is always mass flow. Let me just go through a quick one with you for mass flow. Guys, for mass flow. Oh, this is the calculation question that day. Normally, I'll just use one slide to teach everything that I want to. So have a look. Guys, mass flow, exam come out, let's say they ask you 8 marks. What's your first point? Second point? Third point? Fourth point? Fifth point? This is how you should revise. Guys, this is what you must know. This is what you must know. Check. Okay, this one. So all my processes are in one slide because at the end of the day, in your brain, one picture, one diagram is one unit. Easy to remember. Tak perlu hafal susah payah beratus-ratus unit. Okay, look. First point, during the day, photosynthesis occurs. Sucrose is produced in mesophyll cell. Guys, this is the leaf. So sucrose is produced in the mesophyll cell. They are actively loaded into the sieve tube. They are actively loaded into the sieve tube. Water potential of the mesophyll cells is lowered. Okay. And oh, sorry. The water potential of sieve tube, not mesophyll cell. Water potential of sieve tube is lowered, meaning water potential mesophyll cell is high. Guys, water always move high to low. Water potential. We say that hydrostatic pressure of sieve tube in the leaf region is high. Okay, again, hydrostatic pressure of sieve tube in the leaf region is high. Because later we'll say this is high, this is low, then there's a gradient. This is what causes bulk flow. Okay, guys, again. During the day, photosynthesis occurs, sucrose is produced. It is actively loaded into the sieve tube. Water potential of the sieve tube is lowered. Therefore, water potential of mesophyll cell is high. Water diffuse into the sieve tube. Hydrostatic pressure is high. Next point, guys, what's your next point? If you're very clear, you should be able to tell me, oh, sir, now at the roots. At the roots, oh, this one, not too important, but xylem Water from xylem move into mesophyll cell to replace water loss. Guys, at the roots, sucrose is taken up to be used by the root cells. Now, water potential of sieve tube is high. The root yellow color is low. So water move high to low. Guys, water move out by osmosis. The hydrostatic pressure in the sieve tube of root region is low. Now there exists a high hydrostatic pressure gradient, and this is what causes bulk flow from the leaves all the way to the roots. Guys, this is mass flow. Mass flow means bulk. Sekumpulan. Okay. So yeah, guys. Rather than using the words here, you can just use the diagram earlier. It takes 1 minute only. Guys, it takes 1 minute. And even for my notes, I already simplified. You cannot get it simpler than this. Yeah. Anything simpler than this is you cannot score. And don't forget, guys, for all the processes, the reason why you have so many processes to learn is simply because until today, there's no confirmation. I always tell my students when I was studying STPM, I thought how lucky the future students would be. Because guys, if they accepted mass flow entirely, it means there's no more downside. Sorry, no more evidence against. They're able to prove everything. Then you only study one theory. Even water transport, you only study one theory. Yeah. But until today, don't have. Okay, so for transport, focus on blood circulatory system, these few things, translocation, then only the rest. Looking at chapter 9 now, which is a very long topic. Okay, last year, chapter 9, they asked non-steroid. I do not know about you, but to me, I really like studying adrenaline. Guys, I really like studying adrenaline. Okay, because it's very easy. Very easy. So if I'm a student, I'll feel a bit unfortunate that it came out. How about the previous year? Gibberellin, abscisic acid. Before that, sliding filament. This one, hormones. Okay, hormones. At the end of the session, I'll teach you how to make use of the past year analysis. Okay. Now, for chapter 9, guys, for bab 9, I would say nervous system is more important than hormone. Nervous system is more important than hormone. And for hormone, the two things that you should focus on is steroid, because non-steroid just came out. You can read through one time, no harm. And also phytohormone. Guys, remember long day plan, short day plan, the process? Long day plan, short day plan. Okay, as I said, bab 9 is very long. So which one should I focus on a bit more? Due to the limited time. Resting and action potential, synapse. Sliding filament theory, I worry it comes out as a surprise question. Surprise meaning, yes, it came out 3 years ago, but surprise, guys. And also drug action, these 4. Then only you go through the rest. After you have studied these 4, focus on comparison. They ask you to compare this as well as this. Okay, compare movement of impulse in the synapse, compare movement of impulse in the axon, along the axon. Yeah. So for para and sympathetic, actually it's very easy lah. Got a shortcut. If you remember, I give you one example. Example function, guys, you don't need to study all one. There's a method. For the functions, para is, I call it a brake. Sympathetic, I call it accelerator. It's like driving. Okay, but probably your teacher would have told you is to stop something except for digestion and excretion. Except digestion and excretion. Okay, for example, guys, let's say heartbeat. Heartbeat, is it a digestion? No. Oh, I better write para. Here is para. Here is sympathetic. So heartbeat, is it a digestion? No. Excretion? No. So it will accelerate during sympathetic. If para, then it will slow down. Okay. Another example. Let's say good one would be esophagus. Okay, esophagus or stomach lah, guys, stomach. Okay, stomach, is it an organ for digestion? Yes. It is an organ for digestion. Now, by right should be slowed down, but here there's more digestion. It secretes more enzyme. Yeah, and during sympathetic, it will slow down because opposite, less digestion. Okay, less gastric juice. So that is why when you eat a lot, you tend to be in a para state. Para is resting. Para is resting state. So guys, you can check this out. It's very useful. It's very useful. Last one. Let's say bladder. Let's say urine. Bladder. So para is where urine is stimulated to be released, but not during sympathetic. Not during sympathetic. So everything opposite, guys. Everything opposite. So this is how you should tackle bab 9. This is how you should tackle bab 9. Let me go through one process, the synapse. Okay, guys, we are checking out synapse. Now, synapse, okay, this is the one I'm using. If you're used to it, revision is 1 minute. First point, impulse arrive. Depolarization occurs. The membrane, presynaptic membrane, becomes permeable to calcium ion. Calcium ion enter, causing vesicles to move to presynaptic membrane. Impulse will rupture, releasing neurotransmitters. This will move across, bind to the receptors. Postsynaptic membrane becomes permeable to sodium ion. Sodium ion enters the polarization action potential. If it manages to exceed EPSP, the threshold. This will move along the next neuron. Cholinesterase or acetylcholinesterase will break down the neurotransmitters. Repolarization occurs. Vesicle and acetylcholine form again. Guys, revision 1 minute. If you are seeing this for the first time, maybe you cannot hit, but those who have followed me for quite a while, they have seen this too many times. Revision is 1 minute. Yeah, 12 points. All right, chapter 9 focus on nervous system a bit more for the part of hormones, steroid. process, which is quite short, 6-7 points. Then phytohormone. Okay, for chapter 10, guys, for chapter 10, yeah, chapter 10, okay, last year, what came out for Bug 10? Don't have, right? Yeah, correct, don't have. Guys, last year nothing came out for Bug 10, but probably you can take note of the previous year. Previous year, what they asked was, I'm going to use orange. Okay, seed development, and then adaptation. Previous year also don't have. That's why Bug 10 is kind of like the most important topic. They rarely ask the past few years. They rarely ask. So Bug 10 itself, I want you to draw 2 stars. I mean, if you have this mind map, okay, 3 stars, 3 stars. Why 3 stars? Number 1, it's a very heavy topic. Number 2, never asked before at all last year. You see, every year they show us nearly all the topics except one. Yeah. Okay, sir, I feel it's a very long topic. What should I focus on? Okay, first up would be embryonic development. This is for human, not seed. Just now was seed. Okay, the roles. Hey, there's one more missing. Let me add in. There's one thing missing. Parturition. Parturition. 2 stars. Okay, seed germination, which starts with imbibition and then growth curve. So guys, read these few things before you read the rest. And if they want to ask you surprise question, which many cannot answer, is probably passage of the sperm. Passage of the sperm. Guys, things like fertilization, menstrual cycle, you have already learned since gametogenesis, spermatogenesis, oogenesis, you have learned from Form 4 and Form 5. Okay. Next, for homeostasis, okay, next topic, homeostasis. Okay, homeostasis, last year, oh, the colors, hey, never mind, it's okay. Okay, chapter 11, blood glucose level. I think the previous year is body temperature here lah. So the one that is of lower chance is definitely these two. Liver is number 1. Liver is number 1. Yeah, liver is number 1. And like it or not, no matter what, homeostasis, the importance you have to know. Guys, don't care whether you are whichever year STPM student. Okay, liver, make sure you know the structure and function. Osmoregulation, the pH, that's pH. pH. And for chapter 12, immunity. Chapter 12, immunity. Last year don't have. Okay, last year don't have. But the thing for chapter 12 is that whichever year you are from, these 3 things you must know. It's a very short topic, chapter 12. I taught this in 2 hours, 2.5 hours. The main focus is cell-mediated and humoral. Cell-mediated and humoral. Yeah. Let me just go through cell-mediated with you once. Self and non-self, they can ask. Guys, you check out, I think there was 1 or 2 years they did ask. Essay, yeah, in the form of essay. Is it this? Oh, not this. Wrong slide. Our cell mediator is beside. This is humoral. All right, guys, let's start. So we are looking at cell-mediated response. Okay, first point, macrophage will engulf the pathogen. Pathogen will be broken down and MHC class 2 protein, a major histocompatibility complex protein, will present the antigen on the cell, on the macrophage. Guys, we call this APC, antigen presenting cell. The APC, antigen presenting cell, will release cytokine, which is IL-1, interleukin 1. Helper T cell bind to APC and it is activated. CD4 protein will further enhance the binding. CD4 protein will further enhance the binding. Guys, IL-1 stimulate helper T cell. Helper T cell is activated. It will proliferate into a clone of T cells, including these 4. IL-1 will also stimulate helper T cell to form IL-2. Interleukin 2. Interleukin 2 stimulates cytotoxic cell to proliferate and stimulate these cells to bind to infected body cells to release porphyrin to puncture the infected cell. Once all the pathogen are removed, guys, suppressor T cell suppress action of cytotoxic T cell. This is called primary immune response. This is called primary immune response. Okay, one more time. Okay, so pathogen broken down, MHC class 2 protein will present it on the cell. We call this APC. IL-1 stimulate, CD4 enhance the binding of helper T cell to APC. IL-1 stimulate helper T cell to proliferate into a clone. Then stimulate helper T cell release IL-2, interleukin 2, stimulate cytotoxic T cell proliferate, and also to puncture infected body cells by releasing porphyrin. Okay, when all are destroyed, suppressor T cell suppress action of cytotoxic T cell. Guys, this is called primary immune response. Okay, so you go through secondary, it's very much easier. Secondary is always stronger than primary. Okay, guys, if you realize there's nothing to do with bug 13 here in terms of the mind map. Because you must go through all the 4 diseases: malaria, dengue, tuberculosis. Okay. You see, but last year did come out dengue lah. Maybe dengue is lesser importance, but the rest you have to cover. Okay, the other 4 all you have to cover. Okay, guys. One last thing. This is one last thing. If you are keen to join me in Sem 3, I'll be starting on the 20th. On the 20th of August, right after your math paper. Guys, you have to start somewhere. And as I said, you'll be taking your Sem 1 in Sem 3. Sem 1 retake is in Sem 3. Then Sem 2 retake, ulangan is after Sem 3. I mean, that's what happened the past 2 years. Before the past 2 years, it was really crazy because students had to take Sem 1 and Sem 2 ulangan during Sem 3 itself. Guys, you don't believe, you check with your seniors. So if you are keen to join me in my Sem 3 class, it starts on 20th. Every Tuesday, 5:30 to 7. There'll be recording until you take your Sem 3. And if you have any friends who are Sem 1 students, your juniors, I'm starting on the 7th. Actually, it's tomorrow. Yeah. That's recording, but it's 5 times a month because of the timing. This is 70 per month for both. Okay. So guys, I hope this session did help you. I'll see you when I see you. Okay, good luck. Yeah, good luck. Take care. Bye-bye.